the laboratory techniques, we're going to start with the polymerase chain reaction. It's a microbiology lab procedure used to amplify, so basically creating new exact same DNA. Is just increasing the amount of the DNA that we need of a desired fragment of DNA, a specific sequence of DNA that we want to amplify and study. It's useful as diagnostic tests, just in case a neonate has an HIV or herpes and solitis, just to make sure if this patient has the disease or not. It goes through multiple steps. First is the denaturation, second annealing and elongation. What we do take from the patient sample is double-stranded DNA, and of course it has two different strands. One that is from the 5' end to the 3' end, the other one is on the contrary side, and that is from 3' end to the 5' end. So that's from a patient sample. It does have to go through denaturation first. Is this DNA template that we're using. We add to it a DNA primers and heat stable DNA polymerase and deoxynucleotide triphosphates. They're all heated to close by 95 Celsius degrees to separate the DNA strand. So in step 1, that is the denaturation, we are going to increase the temperature to do one simple thing, and that is the denaturation of this original double-stranded DNA into two separate strands. We already also have a pre-created, something that we have created, primers, DNA primers, plus deoxynucleotide triphosphase. They are going to create a new DNA for each one of those single-stranded DNAs after their denaturation. So that's step 1. Step 2 is the annealing. This sample, the same exact sample, is going to be cooled to nearly 55 Celsius degrees. The DNA primers anneal to specific sequence. So the DNA primers that we have already created in the lab, based on our own knowledge of the disease that we're going to study, just like HIV or encephalitis. So that primer is going to sit on a specific sequence of the original DNA, and then it's going to use this heat stable DNA polymerase enzyme to create a new DNA that's complementary to the original one that we have, and this one is called elongation. As the temperature is increased to nearly 72 Celsius degrees, the DNA polymerase is the enzyme that is going to add these nucleotides, like deoxynucleotide triphosphate. As we see, each one of those is called deoxynucleotide triphosphate. They're going to be added in series to create this complementary DNA to the original one. It replicates the sequence after each primer. They're going to keep moving like that and create this new DNA. This heating and cooling cycle continues until the amount of DNA is sufficient, until we get the enough amount of the same DNA that we're looking for, so we can actually study it and decide if this patient does have such disease or not. CRISPR-Cas9, it's a genome editing tool. So it's basically changing the genome. It's derived from bacteria and it's not used clinically yet. It consists of a guide RNA, that's a specific sequence of RNA that can go to a specific target that we want within the cell. It's complementary to the target DNA sequence, so it's made specifically to be met with that original DNA over here. So this is the original DNA, and it's split into two strands, and this is where the guide RNA comes to be complementary to the target DNA sequence. And also one more thing, it has the endonuclease, that's Cas9, that's what is taken from the bacteria. It makes a single or double strand breaks at the target site. This endonuclease is able to cut the original DNA at specific places. When it does so, these new segments, or like this broken DNA, can be repaired by two mechanisms. One is the non-homologous end joining. That's non-homologous end joining, which means it causes an accidental frame shifting mutation called the knockout, while the other type, and that's 3A. So this is the non-homologous end joining, and as we can see, this is a DNA that has a break within here. So after the non-homologous end joining, there happens a frame shift in activation or a knockout mutation, which leads to a DNA that is lacking something that was original to it, and it was replaced with something new. And this is basically what we want. We want to get rid of the old like diseased DNA that has some genes or alleles of a disease or something, and we want to take it out, so we cut it out, and then it will repair itself, like the DNA will repair itself by non-homologous end joining, or the other type, that is the homology directed repair. So in the HDR, that is a donor DNA sequence can be added to fill the gap. This time we can add, like this, by this other mechanism, is that we intentionally add a piece of DNA called the donor DNA that can come in and fill in like the empty space over here, the space that was cut into two pieces by the endonuclease. So at the end result, we get edited sequence. So it's a knock in. We are actually giving some DNA in to fill that gap. The potential applications include removing virulence factors from pathogens, replacing disease-causing alleles of genes with healthy variants, and that's of course in clinical trials, just like in sickle cell disease, and specifically targeting tumor cells. So the main idea is that we're trying to get rid of something not so good and replace it normally with something that is better and is not diseased. That can be one of four types. The first one is the southern blot. Basically, the main difference between all of these types is this sample type. So in southern blot, the sample is DNA, and in northern blot, the sample is RNA, while in the western blot, the sample is a protein. In the southwestern blot, it's DNA binding proteins. So DNA, RNA, protein, and then at the end, it's a combination between southern and western, so it's a DNA binding protein. That's polymerization. Southern blot is enzymatically cleaved into, there's a DNA sample that is enzymatically cleaved into smaller pieces, which are separated by gel electrophoresis and then transferred to a membrane. So the sample DNA is going to be cut into smaller pieces by enzymes and then transferred to a membrane. In the second step, the membrane is exposed to labeled DNA probes that anneals to its complementary strands. So we have already prepared in the lab a labeled DNA probes that do have the capacity to attach to the strands of DNA from the DNA sample that we have taken. That's 2. 3 is resulting double-stranded labeled piece of DNA is visualized when membrane is exposed to film or digital imager. So because we have already added this labeled DNA probes, so they can be visualized. They are like emitting some kind of light or some kind of color. So whenever they attach to the original DNA sample, then now they create a double-stranded labeled piece of DNA that can be visualized when it's exposed to a film or a digital imager. It's useful to identify the size of a specific sequence, just like the determination of heterozygosity, just like in this image that we're going to get to. And also it could tell us, for example, the number of CGG repeats, that's the repeats within the DNA in the FMR1 gene, to diagnose if this patient has a fragile X syndrome or not. So let's see. So for example, this is the pedigree of a family that has both parents are carrying an allele for some kind of disease, and one of those kids is going to have the disease. So in the southern plots, we're going to use a sample DNA, and then we can separate it out. So the genotype would say which of these is going to be related to it. So here we're going to see the normal alleles, and here are the mutant alleles. So for example, this patient or this kid who has one mutant allele, or it could be just the father. Anyhow, any patient who has one normal allele and one mutant allele would have like two alleles showing, one on the mutant side and the other one on the normal side. If the patient is having like the disease fully, so they don't have a normal allele, they only have the mutant one. And if a patient are actually just normal, doesn't have the disease, then they only have the normal allele and not having the mutant allele. So in order to memorize all of these, there is the snowdrop for memorization. So the southern blot we use the DNA sample, the northern blot will use the RNA sample, while the western blot would read the protein, and the last one is the southwestern blot. It would identify DNA binding protein. So it's a combination between southern and western, okay? The northern blot, it's similar to southern blot, except that's the RNA sample, as we said. It's extrafluorescent, basically the same main idea that we have just used for the southern blot. Useful for studying mRNA levels and size. We know that the level, or like the amount of mRNA that we have within the cell is reflective of gene expression, because messenger RNAs are important for gene expression, and it detects splicing errors. So this northern blot would detect the splicing errors. While the western blot samples the protein, as we said, it is separated via gel electrophoresis and transferred to a membrane labeled antibody. So this time, the probe that we're going to add is a labeled antibody. It's not like a DNA probe, as we said here, or not even an RNA probe, as we said over here in the northern blot. So in the western, we're going to use some sample proteins and add to it some labeled antibody, okay, used to bind relevant proteins, because these antibodies are going to bind those relevant proteins, and this of course helps identify specific proteins and determines a quantity. So they do bind specific proteins and they determine quantity. For the southwestern blot, it identifies DNA binding proteins just like CGen or CFOS, that's leucine zipper motif. Leucine zipper motif is the role in the dimerization of transcription factor, and actually it's much better to take a look. So let's see. Oops. Okay. Church web. And then what is the function of leucine zipper motifs? It's structural motifs typically comprise. Okay, they play a central role in the dimerization of BZIP family of transcription factors and their subsequent binding to DNA promoter regions of the target genes. Okay. Well, of course, the labeled double-stranded DNA probes are to be used over here. So they are not DNA, not messenger RNA, not even antibodies. They are labeled double-stranded DNA probes. Double-stranded. So this one was just labeled DNA probes. Labeled DNA probes, and this time it's labeled double-stranded DNA probes. So southern, that's the DNA, plus western, that's the protein samples. They both have the end results of just polymerization southwestern DNA binding proteins. And that's it for the plotting procedure. So the most important part about it here is that we should know the sample, what is the kind of the sample, and what is the kind of the label that we're going to add to it. And that's it. Techniques are used to assess the size, granularity, and protein expression. So they are mainly asking about the protein. They assess the size, granularity, and protein expression. That's the immunophenotype of individual cells. So they take each cell at a time, not like collection of cells, but each cell at a time within one single sample. So multiple steps, cells are tagged first with antibodies. Those antibodies are specific to surface or intracellular proteins. So what we have like a sample, and that has multiple cells, and each one of those cells has either like surface proteins or intracellular proteins. So what we do is that we give this sample some antibodies that are they're specific to this surface protein, or specific to the intracellular protein, like over here. The antibodies are then tagged. Those antibodies that we have added are then going to be tagged, okay, with a unique fluorescent dye. We're going to color it with a fluorescent dye so we can actually visualize it later. Sample is analyzed one cell at a time by focusing a laser on the dye that we have added over here, and we can actually measure the light scatter and intensity of the fluorescence. And this is quite obvious in this image. So what we have over here is that basically a collection of cells, but we take each cell at a time. So this cell either has like surface proteins or intracellular proteins. This time we're talking about like the surface proteins, like the D3 or a CD3 or CD8. So what we do is that we add to this cell intentionally an anti-CD3 antibody or anti-CD8 antibody. This antibody is going to have, the one that we add is going to have some fluorescent label, okay? So whenever it attaches to the cell and goes in this process where we can actually get to visualize each one of those cells by directing some laser on those cells and detecting how much these cells are going to emit because of this fluorescent label that we have added, we can actually tell which one of those cells is going to express CD3 or CD8, depending on how much antibodies are attached to these cells. So the fluorescence is dissected and labeled cells are counted, okay? And they're going to be visualized on a graph just like this. So the data plotted either on histogram, that's one measure, that's when we measure like one thing at a time, or a scatter plot where we can have like two measures at a time. Just for, as shown in over this illustration, we are detecting the presence of CD3 on the surface and CD8 on the surface of the cells. So we get to visualize which cells have CD3 or CD8. So the cells on the left, so if we take a look at this graph, this side says if it's negative, then the cells that are going to show over here, they are negative of CD8. They don't have CD8. Or they could be positive, so they could have CD8. The same thing goes over here. It's the CD3. So either those cells don't have CD3 or these cells do have CD3. Cells in the left lower quadrant, they are negative for both CD8 and CD3. So this is the left lower quadrant, and we can tell it's negative because over here is negative CD8 and also negative CD3. So the cells that show up on this scatter plot are going to be negative for both of these markers. For the cells in the right lower quadrant, that's this space over here, it's positive for the CD8. So this is the positive side of the CD8, okay? And they are at the same time negative for CD3. So because they're lying over here, it's negative of the CD3. There you go, negative CD3. So it's empty, of course. It's empty because all the CD8, like all the CD8 positive cells, like all the cells that do have CD8, they also have CD3. So they cannot fall over here, okay? Because if they have fell over here, it would be negative CD3. They don't have CD3, but that actually in this example, in this patient, or in these proteins, or in these cells, specifically, whatever cell that has CD8, like positive for CD8, also does have CD3. That's why we see over here, okay? That's why we don't see any cells that are positive CD8 and negative in CD3. Okay, that's why we don't see any cells here. So all of the cells that express CD8 also express CD3. That's why we see them over here. Okay, the cells in the left upper quadrant, that's this space, is positive for CD3 and negative for CD8. Well, makes sense. Over here, it's positive in CD3, okay, but negative in CD8. Right. Cells in the right upper quadrants, that's over here, it's positive for both. Like it's positive for CD8 as over here and positive for CD3 as well. This flow cytometry is commonly used in workup of hematologic abnormalities, just like leukemia, paroxysmal nocturnal hemoglobinuria, fetal RBCs in a pregnant person's blood, and immunodeficiencies like CD4 positive cell counts in HIV, to check on hematologic abnormalities as these examples, and immunodeficiencies. That's CD4 plus cell count in HIV, for example. Is consisting of thousands of DNA oligonucleotides arranged or grids on a glass or a silicon chip. So what we have is this is a chip made of glass or silicon that has like a huge amount, like thousands of DNA oligonucleotides, or it could be RNA. So the sample could be DNA or RNA samples, but they're being compared to each other. They are going to reflect the transcription of the genes. For example, if it was two RNA samples, we're comparing different RNA samples, so we are going to see their rate of transcription within those genes. And the same thing applies for the DNA, if the sample was a DNA. So these DNA RNA samples are compared that are being compared are attached to different fluorophores and hybridized to the array. They are going to be attached to this chemical material that has the ability to re-emit the light. They are also hybridized to the array, so they are going to be mixed with everything else. And the ratio of the fluorescence that comes back to the examiner at a particular oligonucleotide reflects the amount of hybridizing nucleic acid in the two samples. Like this is sample 1 and this is sample 2, and because we are using fluorophores, so we can actually detect the light coming from this hybridization. Some of those oligonucleotide like have a lot of color, like fluorescence coming out of it. On the other side, like the one that we're comparing to, it has only like a few light re-emitting from it. So this light, this comparison between these two are going to lead us to how much transcription is occurring in those genes. It can detect a single nucleotide polymorphism and also copy number variants for genotyping. So we can use it for genotyping, for clinical genetic testing, forensic analysis, cancer mutation, and genetic linkage analysis when DNA is used. So if we are using DNA, then we can also do genetic linkage analysis. With, of course, cancer mutation, forensic analysis, clinical genetic testing, and also genotyping, because it can detect two things. First is the single nucleotide polymorphism, and the second is copy number variants. So that's for microarrays. Immunoabsorbent assay, it's the same as ELISA, and it's very important and very famous, especially in HIV infections. It's an immunologic test. This one is an immunologic test. It's used to detect the presence of either a specific antigen or antibody in a patient's blood sample. So it's either testing an antigen or an antibody. One of those two things in a blood sample. This detection involves the use of antibody linked to an enzyme. So the material that we are going to use, whether we want to detect an antigen or an antibody, we are going to use antibody linked to an enzyme. So that whole entity is going to be used by the examiner, by us. In addition to that, we're going to add a substrate. It's a material that reacts with the enzyme that we have already added, so we can produce some detectable signals. Can have a high sensitivity. Well, the ELISA can have a high sensitivity and specificity, but it is less specific than the Western blot that we have already talked about over here. This is the Western blot, one of the blotting procedures. It's often used to screen for the HIV infection. So it could be on one of two types. It's either direct ELISA or indirect ELISA. For the direct ELISA, we are going to test the presence of this antigen, okay? So we're going to add an entity of enzyme added to a labeled antibody, and if it attaches to that antigen, then it's good. Then we're going to add some substrate. That substrate is able to react to the enzyme that we have added. Now we have a detectable signal. So we can know that this enzyme attached to the antibody was able to attach to an antigen. The indirect ELISA is also going to detect either like antigen or antibody itself. But we are going to add the same entity as we said, okay? And then we may add some other entity, and it's also composed of an enzyme and an antibody. It's called labeled secondary antibody. And over here we can also add this substrate. That's the material that is going to emit some light or some color, so we can actually detect what is happening within the cells, or like within the patient's sample, like patient's blood sample. And over here we can know that there is an antigen as well, and also just an antibody. So that's it for the enzyme-linked immunosorbent assay. That's the ELISA. Colchicine is added to cultured cells that halt the chromosomes in the metaphase. Chromosomes are stained, ordered, and numbered according to the morphology, size, arm length ratio, and banding pattern. Just like the arrows in A point to extensive abnormalities in cancer cells. So this is shape A. These are the doubled arrows over here, and they do show some extensive abnormalities in cancer cells. So over here we have cancer cells. So two things. First, they're going to be the metaphase chromosomes are going to be stained, ordered, and numbered depending on their morphology and size and arm length ratio and banding patterns. It can be performed on a sample of blood, that's one, or a sample of bone marrow, or a sample of amniotic fluid, or a placental tissue. It's used to diagnose chromosomal imbalances, just like autosomal trisomy, sex chromosome disorders. So, for example, over here we do have duplicates of a specific chromosome, so that's why we can tell if we have an autosomal trisomies or just sex hormone disorders. Situ hybridization, there is a fluorescent DNA or RNA probes bind to a specific gene or other site of interest on a chromosome. So mainly we're going to use a fluorescent DNA or RNA probes that are going to bind to either one of two. It's either a specific gene or some other site that we are interested in on the chromosomes. They are used for specific localization of genes, like if we want to detect what is the specific location of that specific gene. And also it's used for direct visualization of chromosomal anomalies, because we can color those so we can tell like what are the abnormalities specifically. So it could be multiple stuff, but mainly microdeletion. We could have microdeletion there. We can tell by seeing that there is no fluorescence on a chromosome compared to fluorescence at the same locus, but on the second copy, because we know that the chromosome has two copies. So in one of those, we don't see a fluorescence at a specific location, while in the second copy we can see that fluorescence. So we can tell that there is microdeletion happening over here. We also it could visualize the translocation. It's as an image A over here is the fluorescence signal from the ABL gene that corresponds to one chromosome. That's on chromosome 9. So over here is the chromosome 9, and this arrow is the fluorescence signal coming from the ABL gene. It's this time found in a different chromosome that it should not be found on, just like chromosome 22, and it's over here. So we can see the same fluorescence signal. This time we can see it's next to the BCR gene, so it's in correct location. So that's for the translocation. Plus we could see the duplication. It's a second copy of a chromosome, resulting in a trisomy or tetrasomy. So there is just a whole new copy of the same chromosome, so we can see a trisomy or tetrasomy as an end result. There is protection of recombinant DNA molecules in a bacterial host. So we're using the bacteria as a place where we can reproduce DNA. It's useful for the production of human proteins in bacteria, just like human growth hormones or insulin, because these two things, for example, as very important, and we would love to create more of those. So we're going to use the bacteria's ability to replicate a lot. So the steps are, first we isolate the eukaryotic messenger RNA. That's the post-RNA processing of interest. So the post-RNA processing or the messenger RNA of insulin, for example, okay? So we do isolate that, and then we add reverse transcriptase, and that's an enzyme, as we know, STASE. So it's an enzyme. It is an RNA-dependent DNA polymerase. So it's a DNA polymerase. It's an enzyme that it creates DNA depending on RNA. That's to produce a complementary DNA. That's why it has a C over here. That's complementary DNA, and complementary DNA lacks introns. Third step is that we insert this complementary DNA that we have created by adding the reverse transcriptase. That is going to be inserted. We're going to insert these complementary DNA fragments into the bacterial plasmids. So it's going to be inserted into the bacterial plasmids containing antibiotic resistance genes. These bacterial plasmids that we are going to implant our complementary DNA fragments in are supposed to be antibiotic resistant genes, because we're going to see at some point we are going to add some antibiotics. So we only need to kill the bacteria, but at the same time we need to protect the DNA that we're creating. So that's the main idea. We are trying to create a DNA, and at some point we would like the bacteria to die, but we still want our DNA to be perfect and protected. That's why we're going to implant it in a specific plasmids that are containing antibiotic resistance genes, okay? So for step 4, we're going to transform, that is, which means insert, the recombinant plasmid into bacteria. So now we are going to use that nice, beautiful antibiotic resistant genes, the plasmid that has this nice quality into a bacteria, okay? Because the bacteria is going to replicate really fast. The surviving bacteria on the antibiotic medium produce cloned DNA because they are surviving. So in the antibiotic medium, like their antibiotic resistance, so they're going to produce DNA, clone DNA. That's the copies of complementary DNA that we're going to use, like as medication or whatever. And that's it. These are the steps of molecular cloning. Type of the laboratory techniques, that's the gene expression modification. It's a transgenic strategies in mice include one of two. It's either random insertion of a gene into a mouse genome, so basically we're inserting a gene into the genome of a mice, or there is a targeted insertion, so it could be random or targeted insertion, or deletion, of course, of a gene through a homologous recombination with mouse gene. So the process is the homologous recombination because it's an insertion, so it needs to be worked on by a process, and that process is homologous recombination. We know that the name knockout should mean removing a gene, so basically taking it out. That's why it's called knockout. Or knock in, just like inserting a gene. If it's a random insertion, then it's called constitutive expression. If it's a targeted insertion, then it's a conditional expression, because we have intended to do this insertion at a specific location. The other type is the RNA interference. So they are types of RNA that do interfere with their replication. It's a process whereby small non-coding RNA molecules, they do target the messenger RNAs to inhibit gene expression. So now we're trying to stop the gene expression. It could be one of two, either microRNA or small interfering RNA. For the microRNA, they are naturally produced by cells as hairpin structures, so they look like hairpins. They lose the nucleotide pairing, so thanks to those microRNA, the nucleotide pairing is not as tight as it's supposed to be, which allows broad targeting of related messenger RNAs. And because of that, it gives us more chance to target broad, like multiple options of the messenger RNA. When the microRNA binds to the normal messenger RNA, or like the functional messenger RNA, this microRNA is going to block this translation of messenger RNA and sometimes facilitate its degradation. So mainly it's going to block its effect, like the microRNA is going to block this translation of messenger RNA, and sometimes it can facilitate its degradation. The abnormal expression of microRNAs could contribute to certain malignancies by silencing an mRNA from tumor suppressor gene. So let's say there is a cell that has a tumor suppressor gene, but its messenger RNA is not functional because it, like microRNA, has attached to it. So now we don't have a controlled cell death. That's how we could develop some kind of malignancies. So that's for the microRNAs. There is another interfering type. It's called small interfering RNA. It's usually derived from exogenous double-stranded DNA. So that's exogenous, and that is double-stranded RNA source, just like a virus from a virus. Once it's inside the cell, this small interfering RNA, it requires complete nucleotide pairing. So that's a must. It's to have complete nucleotide pairing, leading to highly specific messenger RNA targeting. Because of that complete nucleotide pairing, we could have highly specific messenger RNA targeting. It results in messenger RNA cleavage prior to translation. So it would lead to degrading this messenger RNA before it can actually get to work and do the translation. It can be produced by transcription or chemically synthesized for gene knockdown experiments. So we can get it by one of two means. It's either transcription, or it could just be chemically synthesized, and the target, or like the aim that we're going to use it for, is basically gene knockdown experiments. And that's it for the lab techniques.